2001 Nobel Prize in Chemistry. I chose this topic because it shows how a very small difference in structure can lead to a huge difference in function. The prize was awarded for the development of methods in asymmetric synthesis. At first, this may sound difficult, but the idea can be explained with a simple example. Imagine your hands. Your left hand and your right hand look very similar, but they are not the same. They are mirror images of each other. In chemistry, some molecules also have this property. They have the same atoms, but their structures are slightly different, like left and right hands. This difference is very important. For example, in medicine, one version of a molecule can be helpful, while the other version can be harmful. Therefore, scientists need a way to create only the correct version of a molecule. This is where asymmetric synthesis becomes important. Using special catalysts, scientists can control chemical reactions so that they produce mostly one desired form instead of a mixture. It is like having a machine that only produces right-handed gloves instead of both left and right. This breakthrough made it much easier to produce safer and more effective drugs. It also improved efficiency in chemical production, reducing waste and cost. I find this discovery very interesting. It shows that even when two things look almost identical, their effects can be completely different. It also reminds us that precision is extremely important in science. In the future, this technology may lead to the development of better medicines and more environmentally friendly chemical processes. Even though this topic may seem complex, it teaches us an important lesson. Small differences in structure can create big differences in results. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2002 Nobel Prize in Chemistry. I chose this topic because it shows how science can reveal the structure of life by making invisible molecules easier to study. The prize was awarded for the development of methods for the identification and structure analysis of biological macromolecules. At first, this may sound technical, but the main idea is quite easy to understand. Our bodies are made of many important molecules, such as proteins. These molecules are essential for life, but they are extremely small and very difficult to observe directly. Scientists wanted to answer two important questions. First, what kind of molecule is this? Second, what shape does it have? These questions are very important because the function of a biological molecule often depends on its structure. One major breakthrough came from mass spectrometry. This method made it possible to measure large biological molecules more accurately than before. A key breakthrough was soft desorption ionization, which allowed fragile biological macromolecules to be turned into ions and analyzed without being broken apart. In simple terms, scientists could finally identify delicate molecules without destroying them too easily. This opened the door to studying proteins in much greater detail. Another important breakthrough came from nuclear magnetic resonance, or NMR. This method allowed scientists to analyze the three-dimensional structure of biological molecules in solution. In other words, researchers could study not only what a molecule was, but also how it was shaped. That was extremely important because shape often determines function in biology. This discovery had a huge impact on chemistry and life science. It helped researchers understand proteins, disease mechanisms, and many important biological processes. It also supported progress in medicine since scientists could study the structure of molecules related to health and disease. I find this discovery very fascinating. It shows that scientific progress is not only about finding new substances, but also about developing better ways to see what already exists. It also reminds us that understanding life requires both identification and structure. In the future, these methods may continue to support drug development, medical diagnosis, and biotechnology. Even though this topic may seem advanced, it gives us an important message. When we learn how to observe the hidden world more clearly, we can greatly deepen our understanding of life. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2003 Nobel Prize in Chemistry. I chose this topic because it shows how life depends on controlling what can enter and leave cells. The prize was awarded for discoveries concerning channels in cell membranes. At first, this may sound complicated, but the idea is quite simple. Every cell in our body is surrounded by a membrane. This membrane acts like a barrier, separating the inside of the cell from the outside environment. However, cells must exchange substances to survive. For example, nutrients need to enter the cell, and waste products must leave. This is where channels play an important role. Channels are special proteins embedded in the cell membrane. They act like gates that allow specific substances to pass through. One key discovery was aquaporins, special channels that allow water molecules to pass through cell membranes efficiently. Others allow charged particles, such as ions, to move in and out. Another was the structural study of ion channels, which showed how cells selectively control charged particles. This process is highly selective. It is similar to a security system. Only certain molecules are allowed to enter, while others are blocked. This discovery was very important. It revealed how cells maintain balance and control their internal environment. It also helped scientists understand how signals are transmitted in the body. I find this discovery very interesting. It shows that life is not just about structure, but also about controlled movement. It also teaches us that systems function properly only when there is a clear rule for what is allowed to pass. In the future, this knowledge may help develop new medical treatments, especially for diseases related to cell function. Even though this topic may seem complex, it gives us an important message. Life depends on selective control, not just open access. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2004 Nobel Prize in Chemistry. I chose this topic because it shows how living systems maintain balance by removing unnecessary components. The prize was awarded for the discovery of ubiquitin-mediated protein degradation. At first, this may sound complicated, but the basic idea is very logical. In our bodies, proteins are constantly being produced. They play important roles in structure, function, and regulation. However, proteins do not last forever. Some become damaged while others are no longer needed. If these proteins were not removed, they would accumulate and disrupt normal cellular function. This is where the ubiquitin system becomes important. Ubiquitin is a small molecule that acts like a tag. When a protein is no longer needed, ubiquitin attaches to it. This tag signals that the protein should be destroyed. The tagged protein is then sent to a structure called the proteasome, where it is broken down into smaller pieces. This process is highly controlled and selective. It is similar to a recycling system. Only the items marked for disposal are collected and processed. This discovery was very important. It revealed how cells maintain order by removing unnecessary or damaged components. It also helped scientists understand diseases related to protein accumulation. I find this discovery very meaningful. It shows that destruction is not always negative, but can be essential for maintaining balance. It also teaches us that control is not only about creating, but also about removing. In the future, this knowledge may lead to new treatments for diseases such as cancer and neurodegenerative disorders. Even though this topic may seem complex, it gives us an important message. Healthy systems require both creation and removal. Good afternoon, everyone. Today, I would like to talk about the 2005 Nobel Prize in Chemistry. I chose this topic because it shows how molecules can be rearranged to create new substances efficiently. The prize was awarded for the development of the metathesis method in organic synthesis. At first, this may sound complicated, but the idea is quite simple. In chemistry, molecules are made of atoms connected in specific ways. To create new materials, scientists often need to change these connections. Traditionally, this process required many steps and produced unwanted byproducts. However, metathesis offers a more efficient approach. In this method, parts of molecules are exchanged, like swapping pieces between two structures. It is similar to taking two different sets of building blocks and rearranging them to form new combinations. This process is guided by special catalysts that make the reaction faster and more controlled. As a result, scientists can create desired molecules with fewer steps and less waste. This discovery was very important. It improved the efficiency of chemical synthesis and made it easier to design new materials and medicines. It also contributed to more environmentally friendly chemistry. I find this discovery very interesting. It shows that instead of building everything from scratch, we can rearrange existing structures. It also teaches us that innovation can come from changing connections rather than adding new elements. In the future, metathesis may lead to advances in pharmaceuticals, materials science, and industrial chemistry. Even though this topic may seem complex, it gives us an important message. New possibilities can emerge from recombining what already exists. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2006 Nobel Prize in Chemistry. I chose this topic because it explains how genetic information is copied and used in living organisms. The prize was awarded for studies on the molecular basis of eukaryotic transcription. In simple terms, this research explains how information stored in DNA is transferred into RNA. DNA contains the instructions for life. However, DNA itself cannot directly perform most functions in the cell. Instead, its information must first be copied into RNA. This process is called transcription. During transcription, a molecular machine called RNA polymerase reads the DNA sequence. It then creates a corresponding RNA strand based on that information. This RNA can then be used to produce proteins, which carry out most functions in the body. In other words, transcription is the first step in turning genetic information into action. This discovery was very important because it revealed how this process works at the molecular level. Scientists were able to understand the structure and function of RNA polymerase in detail. This helped explain how cells control which genes are turned on or off. I find this discovery very fascinating. It shows that life depends on precise information transfer. It also reminds us that even tiny molecular processes can control complex systems like the human body. In the future, this knowledge may help us develop new medical treatments. For example, it may allow us to control gene expression or treat diseases at the genetic level. Even though this topic may seem technical, it gives us an important message. Life is built on the accurate copying and use of information. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2007 Nobel Prize in Chemistry. I chose this topic because it shows how important surfaces are in chemical reactions. The prize was awarded for studies of chemical processes on solid surfaces. At first, this may sound simple, but it is actually very important. Many chemical reactions do not happen in empty space. Instead, they occur on surfaces. For example, reactions in car exhaust systems happen on the surface of catalysts. These catalysts help reduce harmful gases. Scientists discovered how atoms and molecules behave on these surfaces. They studied how molecules attach, move, and react. This understanding made it possible to design better catalysts. Better catalysts can make reactions faster and more efficient. They can also reduce waste and energy use. This has a major impact on industry. For example, it improves fuel production and environmental protection. I find this discovery very fascinating. It shows that the location of a reaction is just as important as the reaction itself. It also teaches us that small-scale interactions can affect large systems. In the future, this knowledge may help us create cleaner energy and more efficient technologies. Even though this topic may seem technical, it gives us an important message. Where something happens can be just as important as what happens. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2008 Nobel Prize in Chemistry. I chose this topic because it shows how scientists can make invisible processes visible. The prize was awarded for the discovery and development of green fluorescent protein, or GFP. At first, this may sound simple, but it is very powerful. GFP is a protein that glows green under certain light. It was originally found in jellyfish. Scientists discovered that they could attach GFP to other proteins inside living cells. When this happens, those proteins also become visible because they glow. This allows scientists to observe what is happening inside cells in real time. For example, they can see where proteins move and how cells function. Before this discovery, many processes inside cells were invisible. Scientists had to guess what was happening. However, GFP made it possible to directly observe these processes. This changed biology and medicine significantly. It helped researchers understand diseases, cell division, and many other biological systems. I find this discovery very fascinating. It shows that making something visible can completely change our understanding. It also teaches us that observation is a key part of science. In the future, this technology may lead to new medical discoveries and treatments. Even though this topic may seem simple, it gives us an important message. Seeing is a powerful way of understanding. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2009 Nobel Prize in Chemistry. I chose this topic because it explains how genetic information is turned into actual biological structures. The prize was awarded for studies of the structure and function of the ribosome. At first, this may sound complex, but the idea is very important. As we learned before, DNA stores genetic information, and RNA carries that information. However, information alone does not create life. It must be translated into something functional. This is where the ribosome plays a key role. The ribosome is a molecular machine inside cells. It reads the sequence of RNA and uses that information to build proteins. Proteins are essential for almost all functions in the body. For example, they form structures, control chemical reactions, and regulate processes. In other words, the ribosome translates genetic code into real biological action. This discovery was very important. Scientists were able to understand the detailed structure of the ribosome. This helped explain how proteins are built accurately. It also contributed to the development of new antibiotics. Many antibiotics work by targeting bacterial ribosomes. I find this discovery very fascinating. It shows that information must be converted into action to have meaning. It also teaches us that precise translation is essential for life. In the future, this knowledge may lead to new medical treatments and technologies. Even though this topic may seem technical, it gives us an important message. Information becomes meaningful only when it is used to create something. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2010 Nobel Prize in Chemistry. I chose this topic because it shows how scientists can build complex molecules by connecting smaller parts. The prize was awarded for palladium-catalyzed cross-coupling reactions in organic synthesis. At first, this may sound complicated, but the concept is very important. In chemistry, many useful substances are made by combining smaller molecular units. However, connecting these units precisely is not always easy. Scientists developed a method using palladium as a catalyst. This method allows two different molecular fragments to be joined together in a controlled way. Because of this, chemists can build complex molecules step by step. This discovery has had a huge impact. It is widely used in the production of pharmaceuticals, electronic materials, and advanced chemicals. For example, many medicines are created using this technique. It also helps improve efficiency and reduce waste in chemical processes. I find this discovery very fascinating. It shows that creating complex systems requires precise connections between simple parts. It also teaches us that control and accuracy are essential in construction. In the future, this technology may lead to even more advanced materials and medicines. Even though this topic may seem technical, it gives us an important message. Complex systems are built from simple parts connected in the right way. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2011 Nobel Prize in Chemistry. I chose this topic because it changed the way scientists think about order in solid matter. The prize was awarded for the discovery of quasicrystals. At first, this may sound unusual, but it is a very important discovery. For a long time, scientists believed that crystals had to have a repeating atomic pattern. In other words, the structure of a crystal was expected to repeat regularly in space. This idea had been accepted as a basic rule in chemistry and materials science. However, quasicrystals did not fit that rule. They showed a new kind of order. Their atomic arrangement was highly organized, but it did not repeat periodically like an ordinary crystal. This was already surprising, but there was an even more remarkable point. Quasicrystals also showed symmetries that had long been considered impossible in normal crystals. These are sometimes called forbidden symmetries. For example, patterns with five-fold symmetry were thought to be incompatible with true crystal structures. According to the traditional view, such symmetry could not fill space in a regular repeating way. However, quasicrystals proved that solid matter could have order without periodic repetition and still display these unexpected symmetries. At first, many scientists doubted this discovery. It seemed to contradict the established definition of a crystal. However, further experiments confirmed that quasicrystals were real. This discovery was very important. It forced scientists to revise their understanding of crystal structure and solid matter. It showed that nature can create order in ways that go beyond older theoretical limits. I find this discovery very fascinating. It shows that scientific rules are sometimes too narrow to describe the full richness of nature. It also teaches us that when new evidence appears, even basic definitions may need to change. In the future, quasicrystals may help scientists develop new materials with unusual and useful properties. Even though this topic may seem abstract, it gives us an important message. Nature can produce patterns that are both orderly and surprising at the same time. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2012 Nobel Prize in Chemistry. I chose this topic because it explains how cells understand signals from the outside world. The prize was awarded for studies of G-protein-coupled receptors, or GPCRs. At first, this may sound complicated, but the idea is very important. Cells constantly receive signals from their environment. These signals can be light, chemicals, or hormones. However, cells cannot directly understand these signals. They need a system to convert them into internal messages. This is where GPCRs play an important role. GPCRs are proteins located on the cell membrane. They detect signals from outside the cell. When a signal binds to a GPCR, it triggers a series of reactions inside the cell. This process converts an external signal into a cellular response. In other words, GPCRs act as translators between the outside world and the inside of the cell. This discovery was very important. It helps scientists understand how cells communicate and respond. It also has major applications in medicine. Many drugs work by targeting GPCRs. I find this discovery very fascinating. It shows that systems must interpret signals to function properly. It also teaches us that communication requires translation. In the future, this knowledge may lead to better treatments and technologies. Even though this topic may seem technical, it gives us an important message. Understanding signals is essential for meaningful action. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2013 Nobel Prize in Chemistry. I chose this topic because it shows how scientists can study complex chemical systems using computers. The prize was awarded for the development of multi-scale models for complex chemical systems. In chemistry, reactions often occur at the atomic level. To understand them accurately, scientists need to consider quantum mechanics. However, quantum calculations are very complex and require a lot of computational power. On the other hand, classical physics is simpler but less precise. The key idea of this discovery was to combine both approaches. Scientists developed hybrid models that use quantum mechanics for the most important parts of a system and classical physics for the rest. This makes it possible to study large and complex molecules efficiently. For example, these models can be used to analyze how enzymes work in the human body. They also help researchers understand chemical reactions that would be difficult to observe directly. This discovery has had a major impact on chemistry and biology. It has improved our ability to design drugs and understand biological processes. I find this discovery very interesting because it shows that different methods can be combined to solve complex problems. It also teaches us that accuracy and efficiency must be balanced. In the future, these models may become even more powerful and precise. Even though this topic may seem technical, it gives us an important message. Combining different perspectives can lead to better solutions. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2014 Nobel Prize in Chemistry. I chose this topic because it shows how scientists overcame a fundamental limitation in observing the microscopic world. The prize was awarded for the development of super-resolved fluorescence microscopy. For a long time, scientists believed that optical microscopes had a fixed resolution limit. This limit, known as the diffraction limit, prevented researchers from observing structures smaller than about 200 nanometers. As a result, many important processes inside cells could not be seen clearly. However, this discovery broke that limitation. Scientists developed techniques that allow imaging beyond the diffraction limit. One method uses fluorescent molecules that can be switched on and off. By controlling these molecules and detecting their positions precisely, scientists can reconstruct high-resolution images. Another approach improves resolution by selectively activating small groups of molecules at a time. These techniques make it possible to observe structures at the nanoscale. This has greatly advanced biological research. Scientists can now observe how proteins interact inside living cells in real time. This discovery has opened new possibilities in medicine and life sciences. I find this discovery very fascinating because it shows that even fundamental limits can be overcome. It also teaches us that creative thinking can redefine what is possible. In the future, these techniques may become even more powerful and widely used. Even though this topic may seem technical, it gives us an important message. Limits are not always absolute. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2015 Nobel Prize in Chemistry. I chose this topic because it explains how living organisms maintain the accuracy of their genetic information. The prize was awarded for mechanistic studies of DNA repair. DNA carries the genetic instructions necessary for life. However, DNA is constantly damaged by various factors. These include radiation, chemicals, and even normal cellular processes. If this damage is not repaired, it can lead to mutations. Mutations may cause diseases such as cancer. Fortunately, cells have developed several repair systems to correct DNA damage. One important mechanism is base excision repair. This system removes damaged bases and replaces them with correct ones. Another mechanism is nucleotide excision repair. It removes a larger section of damaged DNA and rebuilds it. There is also mismatch repair, which corrects errors that occur during DNA replication. These systems work together to maintain genetic stability. Without them, life would not be able to sustain itself. This discovery has had a major impact on biology and medicine. It has improved our understanding of how diseases develop. It also helps in developing treatments, especially in cancer therapy. I find this discovery very fascinating because it shows that biological systems include built-in error correction. It also teaches us that maintaining accuracy is essential for survival. In the future, this knowledge may lead to new medical advances. Even though this topic may seem complex, it gives us an important message. Correcting errors is just as important as creating information. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2016 Nobel Prize in Chemistry. I chose this topic because it shows how scientists can build machines at the molecular level. The prize was awarded for the design and synthesis of molecular machines. A molecular machine is a system made of molecules that can perform controlled movements. At first, this idea may seem unusual. When we think of machines, we usually imagine large devices like engines or robots. However, scientists have succeeded in creating machines that are only a few nanometers in size. These molecular machines can rotate, move, or change shape in response to external stimuli. For example, some molecular structures act like switches that can turn on and off. Others behave like tiny motors that rotate in a specific direction. These movements are controlled by light, heat, or chemical energy. This discovery is important because it represents a new stage in chemistry. Instead of only studying molecules, scientists can now design systems that perform functions. This opens up new possibilities for nanotechnology. In the future, molecular machines may be used in medicine, such as targeted drug delivery. They may also lead to the development of new materials and technologies. I find this discovery very fascinating because it shows that even at the smallest scale, systems can be engineered. It also teaches us that function can emerge from carefully designed structures. In the future, this field may grow and lead to practical applications. Even though this topic may seem futuristic, it gives us an important message. Small systems can perform powerful tasks. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2017 Nobel Prize in Chemistry. I chose this topic because it shows how scientists can observe biological structures at near atomic resolution. The prize was awarded for the development of cryo-electron microscopy. For a long time, it was difficult to determine the structures of large biological molecules. Traditional methods, such as X-ray crystallography, required crystals, which are hard to obtain for many proteins. Another method, electron microscopy, often damaged samples due to high-energy electron beams. Cryo-electron microscopy solved these problems. In this technique, biological samples are rapidly frozen at very low temperatures. This preserves their natural structure without forming ice crystals. Scientists then use electron beams to capture images of the molecules. By combining many images and processing them with computers, they can reconstruct highly detailed three-dimensional structures. This allows researchers to study proteins, viruses, and other complex molecules in their natural state. This discovery has had a major impact on biology and medicine. It has improved our understanding of how molecules function in living organisms. It also plays an important role in drug development and disease research. I find this discovery very fascinating because it shows how technology can reveal structures that were once invisible. It also teaches us that preserving natural conditions is essential for accurate observation. In the future, cryo-electron microscopy may become even more powerful and widely used. Even though this topic may seem technical, it gives us an important message. Seeing structure leads to understanding function. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2018 Nobel Prize in Chemistry. I chose this topic because it shows how scientists can harness the power of evolution for practical purposes. The prize was awarded for the directed evolution of enzymes and for the phage display of peptides and antibodies. In nature, evolution occurs through random mutations and natural selection. This process takes a very long time. However, scientists found a way to speed it up in the laboratory. This method is called directed evolution. In this process, researchers introduce random mutations into a gene that encodes a protein. They then select the variants that show improved performance. By repeating this cycle, they can create proteins with new or enhanced functions. This technique has been used to develop enzymes that work more efficiently or under unusual conditions. It is widely used in industry, such as in the production of biofuels and pharmaceuticals. Another important method is phage display. This technique uses viruses that infect bacteria to evolve proteins that bind to specific targets. It has been particularly useful in the development of therapeutic antibodies. This discovery has had a major impact on chemistry, biology, and medicine. It shows that evolution can be guided and controlled. I find this discovery very fascinating because it demonstrates that natural processes can be turned into engineering tools. It also teaches us that iteration and selection are powerful methods for improvement. In the future, directed evolution may lead to new technologies and medical treatments. Even though this topic may seem complex, it gives us an important message. Improvement can be accelerated through selection. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2019 Nobel Prize in Chemistry. I chose this topic because it is closely related to technologies that we use in our daily lives. The prize was awarded for the development of lithium-ion batteries. Lithium-ion batteries are widely used in smartphones, laptops, and electric vehicles. They have become an essential part of modern society. Before lithium-ion batteries were developed, energy storage systems were less efficient and heavier. This made portable electronic devices difficult to design and use. The development of lithium-ion batteries was made possible by combining several important discoveries. First, scientists developed a cathode material that could store lithium ions effectively. This allowed the battery to generate a high voltage. Next, researchers improved the anode by using materials that could safely store lithium ions during charging. Finally, the combination of these materials created a rechargeable battery that is both lightweight and powerful. One of the most important features of lithium-ion batteries is their ability to be recharged many times without significant loss of performance. This makes them ideal for long-term use in electronic devices. Another advantage is their high energy density, which means they can store a large amount of energy in a small space. Because of these properties, lithium-ion batteries have enabled the development of portable electronics and electric vehicles. They also play an important role in renewable energy systems. For example, they can store energy generated by solar panels and wind turbines. This helps reduce our dependence on fossil fuels. I find this invention very important because it supports a more sustainable future. It shows how chemistry can contribute directly to solving global energy problems. In the future, further improvements in battery technology may lead to even more efficient and environmentally friendly energy storage systems. Even though we use these batteries every day, we often do not think about the science behind them. This award reminds us that scientific innovation can have a direct impact on our lives. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2020 Nobel Prize in Chemistry. I chose this topic because it represents a revolutionary tool that allows us to directly edit genes. The prize was awarded for the development of CRISPR-Cas9, a method for genome editing. CRISPR-Cas9 is a technology that allows scientists to change DNA in a precise and efficient way. DNA contains the genetic instructions for all living organisms. By modifying DNA, scientists can potentially change how cells function. Before this technology was developed, gene editing was difficult, slow, and expensive. CRISPR-Cas9 made it much easier and more accessible. The system works like molecular scissors. It uses a guide RNA to find a specific sequence in the DNA. Once the target is found, the Cas9 enzyme cuts the DNA at that location. After the cut is made, the cell repairs the DNA. During this repair process, scientists can add, remove, or change genetic information. This allows precise control over genetic sequences. CRISPR-Cas9 has many important applications. For example, it can be used to study gene function by turning specific genes on or off. It also has potential in medicine, such as treating genetic diseases. In agriculture, it can be used to create crops that are more resistant to pests or environmental stress. Despite its advantages, this technology also raises ethical concerns. For example, editing human embryos could have long-term effects that are not fully understood. Therefore, careful regulation and discussion are necessary. I find this discovery very important because it gives us powerful control over life at the molecular level. It shows how chemistry and biology can work together to create new technologies. In the future, CRISPR may lead to breakthroughs in medicine and biotechnology. Even though it is a relatively new technology, it has already had a major impact on science. This award reminds us that scientific tools can change the way we understand and interact with life itself. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2021 Nobel Prize in Chemistry. I chose this topic because it introduces a new and powerful way to control chemical reactions without using metals. The prize was awarded for the development of asymmetric organocatalysis. Catalysts are substances that speed up chemical reactions without being consumed. They are essential in producing many important materials, including medicines and industrial chemicals. Before this discovery, most catalysts used in asymmetric synthesis were either metals or enzymes. Metal catalysts can be very effective, but they are often expensive and may cause environmental problems. Enzymes are highly selective, but they can be sensitive to temperature and other conditions, making them difficult to use in some situations. The development of organocatalysis provided a new alternative. Organocatalysts are small organic molecules that contain elements such as carbon, hydrogen, nitrogen, and oxygen. These catalysts are stable, easy to handle, and do not rely on rare metals. A key feature of this method is its ability to create molecules in a specific three-dimensional arrangement. This is important because many molecules, especially in medicine, can exist in different forms that have different effects. In some cases, one form of a molecule can be beneficial, while another form can be harmful. Asymmetric organocatalysis allows chemists to selectively produce the desired form of a molecule. This improves both the efficiency and safety of chemical production. This method has been widely applied in pharmaceutical development, where precise molecular structure is crucial. It has also contributed to more environmentally friendly chemical processes by reducing the need for heavy metals. I find this discovery very important because it provides a simpler and greener approach to chemical synthesis. It shows that small organic molecules can perform complex catalytic functions that were once thought to require metals or enzymes. In the future, organocatalysis may lead to the development of new drugs and sustainable technologies. Even though catalysts are not always visible in final products, they play a central role in modern chemistry. This award reminds us that innovative ideas can transform fundamental processes in science. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2022 Nobel Prize in Chemistry. I chose this topic because it introduces a simple and powerful approach to building molecules, often described as click chemistry. The prize was awarded for the development of click chemistry and bioorthogonal chemistry. In traditional chemistry, synthesizing complex molecules often requires many steps and can be time-consuming and inefficient. Scientists have long searched for methods that allow molecules to be assembled quickly and reliably. Click chemistry provides such a method. It refers to a set of reactions that are simple, fast, and highly efficient. These reactions work under mild conditions and produce minimal unwanted byproducts. A key feature of click chemistry is that it allows small molecular units to click together like building blocks. One of the most important examples is the reaction between an azide and an alkyne, which forms a stable connection. This reaction is highly selective and works well in many different environments. Building on this concept, scientists developed bioorthogonal chemistry. Bioorthogonal reactions are chemical reactions that can occur inside living organisms without interfering with natural biological processes. This is extremely useful for studying biological systems. For example, researchers can label specific molecules inside cells and track their behavior in real time. This allows scientists to observe processes such as protein interactions and cellular activity. Bioorthogonal chemistry has also contributed to medical research, including drug development and diagnostic techniques. It enables precise targeting of molecules within the body. I find this discovery very important because it simplifies complex chemical processes. It transforms the way chemists design and construct molecules. Instead of complicated procedures, chemists can now use reliable and predictable reactions. This approach also promotes greener chemistry by reducing waste and improving efficiency. In the future, click chemistry and bioorthogonal methods may lead to new advances in medicine, materials science, and biotechnology. Even though these reactions are simple in concept, their impact is profound. This award reminds us that simplicity can lead to powerful innovations in science. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2023 Nobel Prize in Chemistry. I chose this topic because it focuses on nanotechnology and how tiny materials can produce powerful and useful effects. The prize was awarded for the discovery and synthesis of quantum dots. Quantum dots are extremely small particles, typically only a few nanometers in size. A nanometer is 1/1,000,000th of a meter, which is far smaller than what we can see with the naked eye. At this scale, materials behave differently from their bulk forms. One of the most important features of quantum dots is that their optical properties depend on their size. This means that by changing the size of the particle, scientists can control the color of light it emits. Smaller quantum dots emit blue light, while larger ones emit red light. This size-dependent behavior is a direct result of quantum mechanics. Before this discovery, controlling the color of emitted light with such precision was very difficult. The development of reliable methods to produce quantum dots was a major breakthrough. Scientists were able to create particles with uniform size and stable properties. This made it possible to use quantum dots in practical applications. Today, quantum dots are widely used in display technologies, such as televisions and monitors. They provide bright colors, high contrast, and energy efficiency. Quantum dots are also used in biological imaging. For example, they can be attached to molecules inside cells, allowing scientists to track biological processes. In addition, they have potential applications in solar cells and medical diagnostics. I find this discovery very fascinating, because it shows how controlling matter at the nanoscale can lead to entirely new properties. It demonstrates the power of combining physics and chemistry at very small scales. In the future, quantum dots may lead to further innovations in electronics, medicine, and energy technology. Even though they are extremely small, their impact on technology is very large. This award reminds us that working at the smallest scale can produce the most colorful and useful results. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2024 Nobel Prize in Chemistry. I chose this topic because it shows how scientists can now both predict protein structures and design entirely new proteins. The prize was awarded for computational protein design and protein structure prediction. At first, this topic may sound very technical, but the basic idea is clear. Proteins are essential molecules in living organisms. They act as enzymes, signals, transporters, and structural components. Their function depends strongly on their three-dimensional shape. For many years, one of the great challenges in chemistry and biology was to understand how a protein sequence determines its structure. One part of the prize concerned protein structure prediction. Demis Hassabis and John Jumper developed methods that made it possible to predict the three-dimensional structure of proteins from their amino acid sequences with remarkable accuracy. This solved a problem that scientists had struggled with for more than 50 years. As a result, researchers can now understand protein structures much faster than before. This has greatly helped biology, medicine, and drug discovery. The other part of the prize concerned computational protein design. David Baker developed computational methods for creating entirely new proteins that did not previously exist in nature. In other words, scientists are no longer limited to studying natural proteins. They can now design proteins with new shapes and new functions. This is a major breakthrough. It means that proteins can be engineered as practical tools. For example, designed proteins may be used in medicine, vaccines, catalysis, and environmental technology. Together, these discoveries were extremely important. Protein structure prediction helps us understand life's molecular tools, while computational protein design allows us to create new ones. I find this Nobel Prize very fascinating. It shows that science is moving from observation to creation. It also teaches us that once we understand structure deeply enough, we can begin to design function. In the future, these methods may lead to new medicines, better enzymes, and innovative materials. Even though this topic may seem advanced, it gives us an important message. Understanding nature can eventually give us the power to build new forms of it. Thank you. Good afternoon, everyone. Today, I would like to talk about the 2025 Nobel Prize in Chemistry. I chose this topic because it represents a new way of designing materials at the molecular level. The prize was awarded for the development of metal organic frameworks, also known as MOFs. MOFs are materials made by combining metal ions with organic molecules. These components form highly ordered, three-dimensional structures. One of the most important features of MOFs is their extremely high porosity. They contain a large number of tiny pores within their structure. As a result, MOFs have a very large surface area compared to their size. In fact, just a small amount of MOF material can have a surface area equivalent to several football fields. This unique property allows MOFs to store and separate gases efficiently. For example, they can capture carbon dioxide from the air. This makes them useful in addressing environmental problems, such as climate change. MOFs can also be used to store hydrogen, which is important for clean energy technologies. In addition, they can selectively separate gases in industrial processes. Another important application is water production. Some MOFs can absorb water vapor from dry air and release it as liquid water. This is especially useful in desert regions where water is scarce. The design of MOFs is highly flexible. By changing the metal ions and organic linkers, scientists can create materials with specific properties. This means that MOFs can be tailored for different applications. I find this discovery very interesting because it shows how chemistry can be used to design functional spaces, not just substances. It represents a shift from studying materials to engineering them. In the future, MOFs may play an important role in solving global challenges. These include energy storage, environmental protection, and resource management. Even though they are made of simple components, their structures are highly sophisticated. This award reminds us that controlling structure at the molecular level can lead to powerful new technologies. Thank you.